have a brief discussion on animal development. Um, this subject is very complex, and a lot of what we know is coming from sea urchins, which of course are deuterostomes, so presumably having sort of the same pattern of development as us, and chicks, which they're birds, of course, and or reptiles, and they would also have the same pattern. So those are the areas of study, not so much what happens in humans, although some things we know from human actual research. So there are four basic points of animal development. There's fertilization, then cleavage, gastrulation, and organogenesis. So we're going to talk a little bit about each of these, but this chapter is very convoluted and there's a lot to know. So really, this is the bottom line. I want you to know a little bit about each one of these things, basically kind of definition form of what these things are and a few facts, but other than that, you don't have to know all the information in that chapter. If you go on to animal development, like if that's an interest of yours, you'll hear a lot more about this. And it's, like I said, a whole course could be made on this particular subject. So first, let's talk about fertilization. So remember that the sperm and egg are meeting up in the oviduct, and the sperm head, which of course has the sperm nuclei that is part of it, so this is basically the sperm head. Of course, this part is the flagella. Basically what is happening, there has to be a certain number, somewhere on the order of, I think it's 10,000 sperm at least, in order for this process to occur. So you have to have all of these sperm trying to get in an egg at the same time for this cascade of chemical reaction that is happening to the outside of the ovum, the wall, basically. So it's not really like a cell wall, but there are materials that are on the outside of the ovum that keep any sperm from going through. Otherwise, you would have 10,000 possibilities of sperm going through. So you have to have one lucky winner, so to speak. And what's going to happen is, as the sperm starts trying to get through, it produces parts. It's called an acrosomal reaction, and that reaction is going to start breaking down or causing a reaction that is inside the egg cytoplasm, and eventually that will break through that barrier and allow the sperm head to make it through so that the nucleus of the sperm can go through. Once that sperm gets through, it basically shuts down the outside or the cytoplasmic envelope to any other sperm making it through. So this is a process that only allows one sperm to get into one egg so that you can have one fertilization and a zygote that is 2N. Remember, sperm and egg are 1N each, and combined they are 2N. All right. So that is the kind of a general thing about fertilization. The next thing that I want to tell you about is cleavage. The first thing that happens is that the body axis is forming on the actual zygote. It's been studied. This is a chick one that they're talking about. And if you look at this figure and sort of, look at it closely, you can see that the body axes are kind of established early on in, just the single cell. And then later, when you actually have cleavage, then that's where the cleavage furrows actually occur between the cells that are doing the division. Of course, the zygote will undergo the first cell division, and then the second one, and then the third one become the 8-cell stage, and then it really starts getting some differences and becomes the gastrula. Now, we talked about the gastrula a little earlier in the semester. I'm looking for that overhead, which I don't seem to have right now. All right, gotta shut this off.